Hi everybody, welcome to another amazing ASGCT 2026 Soundbites episode. I'm your host, Dr. Casy, and joining me today is the amazing Dr. Defne Amado, and she's to talk to us about her work, her career, pretty much, and also what gets you excited. Dr. Amado, how are you? How's the conference going for you so far?
This conference has been absolutely amazing. I'm delighted to be here. Thanks for having me.
Awesome. So you've recently won an award from the organization. I want you to tell us what it is and what got you excited once you found out about it.
Yeah, so this is a Career Development Award, which is a huge honor. And it's based on work that I've developed to control the expression of therapeutics in cell types that are affected by diseases. So I'm really excited about that. I can tell you a bit about how it fits into the larger context of my research. So I'm a physician scientist, and I see people living with ALS. And I study ALS in the lab. I work on figuring out kind of the pathobiology that underlies the disease, and I use AAV-mediated gene therapies to develop therapeutics for that disease. I work very closely with Bev Davidson, who has amazing capsids that can target the parts of the nervous system that are affected in a really specific and strong way. And so the really exciting thing about these therapies is that we're able to deliver them in a very strong way that targets the motor neurons, which are the cells that are affected and dying in ALS patients. And this is what causes paralysis and eventual death in this population. We're able to deliver our therapies, and one of the therapeutics that we developed, which is a microRNA that targets the underlying pathology, extends the lifespan of a mouse model of ALS by 50%. So we're really excited about that. We're working hard to bring that towards clinical translation. There's a lot of other ways that we're targeting the underlying pathology that could work either individually or in synergy to cure this disease. One of the problems, though, is that in a mouse model of disease, every cell has the problem. And so when you deliver your therapy, you're doing something helpful. But in a human, you have healthy cells and unhealthy cells right next to each other. So if you deliver a therapeutic to that population of cells, you might actually be doing harm in the healthy cell that didn't need that therapy to be there. And so what we've devised is a way to deliver the therapy everywhere, but only have it turn on in the cells that are affected by the disease. And in fact, it's so fine-tunable in theory. I mean, the award is going to help us develop this, but it should be so fine-tunable that when the cell gets healthy, it should turn itself off. So that's the idea behind our award.
That's awesome. So you received the email that said you will receive the award. What was your reaction? I want you to take us to that day.
When I applied for the award, I was like, oh, I really want this because I've been a long time believer in ASGCT's mission. I've been a part of ASGCT since I was a graduate student. I trained with Jean Bennett for my PhD, and that was my first gene therapy conference back in 2008. And I actually got a presidential symposium talk at that time as a grad student. And so I've always just been really moved by ASGCT's mission, and I've believed in gene therapy since my very first scientific endeavors. So I wanted to kind of be a part of ASGCT's story and be, a part of the culture moving these gene therapies forward, because I think they're so exciting, and they're the way that we're going to cure so many diseases, including of the central nervous system, which are so hard and refractory to treatment right now. We don't have a cure for any neurodegenerative disease, and I think this is the way that we'll get there. So I was really excited to apply, but of course the odds are low. These are not...
It's not guaranteed.
Are not guaranteed, and so when I got that email, I opened my inbox, and then I left my office and I did a sprint up and down the hall. Just by myself, I just sprinted up and down the hall. And then actually 3 doors down from me is Bev Davidson's office, and I knocked on her door and I told her, and we did a little screaming.
Yeah,
so, yeah.
That's awesome. That's awesome. I can feel the enthusiasm. What advice do you have for postdocs or for people, who are going through the process? Any advice for them?
Yeah, I think one of the things that I find very challenging in the field of gene therapy, in the field of ALS, and these are fast-moving fields that are, it can feel competitive. It can feel like a lot of people are working in a shared space. And if you just reach out to the people that you think are scary and collaborate, say, hey, I'm working on this. I heard you're working on that. I'm really impressed with your work. Is there, synergy here, or could we borrow your cell line? And that approach has been just really fruitful and fun to be part of a team that's working on a disease rather than anyone against each other. I also think finding a good story, sinking your teeth into it, and just sticking through the challenges to persist in getting something across a finish line, even when it's difficult. I think that science takes endurance, it takes resilience, it's mostly failure. It's not you, it's science. If things aren't working, just change it, hammer at it. You have to know when to break up with the project if it's not going to get there. But I think so much of it is resilience and persistence. And so many of the therapies that were successful were a matter of back to the drawing board, change this parameter, try it again. Our own published work in that ALS mouse model, was 7 years of work. Wow. It's a lot of persistence, but it works now. So
that's awesome. So what drew you to the field? Is it like personal stories? Like, I want to know.
Do you mean the field of gene therapy or ALS?
to ALS in particular.
Yeah. So I had an interesting road here. I realized during graduate school that because science was so much failure, I could get demotivated. I could get discouraged. And it was projects that I felt were solving a really important problem, where the motivation to keep going exceeded the motivation to stop going. So in my neurology residency, I always thought the brain was the coolest organ, and I was a neuroscientist from the jump, but I was really looking for what is the disease that would keep my feet pointed back toward the lab that really shouldn't exist. And I met my first ALS patient, and I was like, what the heck is this? This is horrible. It shouldn't exist, and I will be forever motivated to fight it until it goes away. And so that's how I ended up working in ALS. And the truth is that I treat a uniformly terminally ill population. That's really, really motivating to keep running back to the lab and be like, guys, we got to get this right. We have to help these people.
Yeah, that's awesome. I have so many questions to ask you. I want to ask you this one. How do you balance being in the clinic as well as running a research program? But that's a lot.
It's really difficult, and I feel like I have 30 jobs. I'm also a mother of two amazing kids, and sometimes it is hard or impossible to do all of it well all the time. But I do feel that I get really important things from each part of my job. So from my patient population, I develop interesting questions. Hey, the people in clinic really care about this, or I'm noticing this clinically. How can that relate to what we're seeing in the lab? In the lab, I am motivated by those clinical questions, and the things that I learn in the sciences and at the conferences, I can bring back to my patients. Say, hey, I heard this really cool thing at ASGCT. That's interesting, and I'm teaching my patients all the time, and they're teaching me. They bring research to my attention and vice versa. So I actually have a pretty scientific clinic. I like to talk about my science with my patients, and they appreciate that I'm working on their disease. I also, when I'm having a really hard day in the lab, it helps the next day to do something that's immediately gratifying. When I see my patients, I'm helping them right then and there. But because I can't offer them a disease-modifying treatment right now or a cure, I know from this, the sadness of that situation, at least I am working on the problem. So it kind of fulfills all of the ways that I think about things and all of the needs that I have as a scientist and clinician to feel like I'm doing something important.
Yeah. So as of right now, do you feel like you have all the support that you need, or do you think I think you can always use some more support, right?
That's, oh my God. I think that right now the biggest challenge, so I'm junior faculty, I'm trying to launch my lab. NIH funding has dropped dramatically. And it's been very, very hard to obtain, to get timely review. Everything's super delayed. They're funding way less than they used to, and I'm worried about the future of our lab. The Career Development Award is super, super helpful in helping us keep momentum and take on high-risk projects that are high reward. But I think we are going to need to be partially supported by things like philanthropy, by foundation funding. But the truth is that all of those things constitute a fraction of what, an R01 provides for a lab. And so we're kind of trying all avenues to keep our momentum going and keep things moving forward.
Yeah. What are some of the challenges that you foresee, like that you'll be facing once you are ready to take your work into the clinic?
Oh, yeah. Well, I think, AAV therapies, I really have always believed in them because it's a one-and-done therapy. I think for a population like ALS, that's very valuable because it's hard for a patient to come back for monthly injections into their spinal fluid for like ASO treatments or other treatments. AAVs are one and done. So the potential for that, to deliver a therapy that lives in your body and acts forever is huge, is high. On the other hand, right now, AAV therapies can't be turned off once they're delivered. They are risky, therefore, because whatever you're doing permanently. And so there's a lot of, regulatory stuff around that, I think appropriately. But I think that we're going to have to do very careful safety studies. Our lab is working, including on the subject I told you about for our Career Development Award, on fine-tuning the expression of those things and making them safer, making them more targeted, and hopefully that helps us get things across the finish line. But taking a therapy to clinic is very, very expensive. We're definitely going to partner with industry when the time comes to move this forward. I think another challenge is that when, you kind of, right now the way things are structured, you kind of get one shot, right? And then the therapy was a failed trial if it doesn't work, when maybe all it needed was to substitute a different promoter or do this other thing. But right now the trials are so expensive and such a huge undertaking that, investors are skittish after that. So it's a challenge, I think, in the field and for people in academia who are like, no, we just need to do this and it'll work. So I don't know how that's going to go when we get there, but I think we just make things as safe and effective as we can in the preclinical world and then hope for the best.
That's awesome. Well, Dr. Amado, I'm very excited for you. I'm very happy for you. And I look forward to following your career very closely. Thank you for stopping by.
Thank you so much for having me. It's been a genuine pleasure.